Hi, in this video we are solving and understanding the concepts behind question 3 of GATE Biotechnology 2021 paper. The question is, the number of unrooted trees in a phylogeny of 5 sequences is option A 3, option B 15, option C 105 and option D 945. So before we solve this, let's try to understand about phylogenetic trees. The evolution of new organisms is driven by diversity in their genomic sequences arising due to mutations. Phylogeny is the study of evolutionary history of a kind of organism. So, in phylogenetic studies, the most convenient way to study the evolutionary relationship of organisms or sequences is through the illustration of a phylogenetic tree. It is important to study evolution for several reasons, like to understand lineage of several species, to develop an understanding of when and which species and event may have occurred to give rise to the organism as seen today. It's also useful to understand the evolution of functional relationships, to understand evolution of pathogen strains for vaccine development and diagnosis. Like for example, with COVID-19, we had different strains like Delta and Omicron, and it was important to understand the lineage, to understand its pathogenicity, and to develop the vaccine. So, the study of evolution is also used to identify the conserved sequences across species. For example, functionally important coding gene sequences maintain the same structure across different species. So, like I mentioned before, the most practical application is in epidemiology to study disease causing pathogen evolution. So, historically, comparison of morphological similarities and differences have been used to construct evolutionary trees. In this approach, the species or organisms that share certain characteristics, that is, homologous traits like the number of limbs and the structure of the limbs, tend to be placed together on the tree. However, advancement of technologies and understanding allows us to use molecular features like gene or protein sequences. When comparing homologous genes in different species, sequences from closely related species are more similar to each other than sequences from distantly related species. So here are some basic terminologies used in phylogenetics. Leaves or nodes represent the things that are being compared. They could be genes, sequences, samples of individuals, or strains or species, or they could be anything that we wish to compare. So the term taxon is used to refer to this, and its plural is taxa. And internal nodes are hypothetical ancestral units from which descendants branch out. So a root represents a common ancestor from which everything else is evolved. So in a rooted tree, path from the root represents the evolutionary path. So an unrooted tree only specifies the degree of relationship among the taxa but nothing about the evolutionary path, and we will look more into detail about the types of phylogenetic trees in the further slides. So here are some examples of phylogenetic evolutionary trees. It shows the evolution of primates and where humans stand in this evolutionary tree. So what sequences are used to construct phylogenetic trees? So to answer that, nucleotide sequences, that could be DNA or RNA, and protein sequences could be used to construct phylogenetic trees. So DNA is very sensitive and has non-uniform mutation rates. The cDNA or the complementary DNA, that is the sequence complementary to an RNA sequence, or the RNA sequences itself, could also be used to construct trees, and they're used to study more remote homologies. So protein sequences are useful to study the most remote homologies, that is, they could be used to study the evolutionary relationships of very distant species as well. Also, there's one more thing called ribosomal RNA 16S sequence. So for evolutionary studies, we more often see that these ribosomal RNA 16S sequences are used. So 16S rRNA is a component of the 30S subunit in prokaryotic ribosomes, while 18S rRNA is a component of the 40S subunit in eukaryotic ribosomes. So ribosomal RNA or rRNA is a structural component of ribosomes. So rRNA is involved in the protein synthesis mechanism, and it's found in like almost every organism. So, like, why are these sequences used to study evolution again? It's because they exist in all organisms and are highly conserved and hence suitable for broad and deep phylogenetic studies. A phylogenetic analysis consists of a few major steps. So, the first one is to retrieve sequences. So, we saw that we could use either nucleotide or protein sequences. So, the first step is to retrieve those sequences. And the next step is sequence alignment. So, this is the essential and preliminary step where multiple sequences are aligned and scored. So, pairwise distances are then computed and the substitution model is determined. So, that's the next step. And following that, we build the tree, the phylogenetic tree is constructed, and then the reliability of the tree is estimated using certain methods, and then finally the constructed tree is visualized and analyzed. So there are two types of phylogenetic trees. They could either be rooted or unrooted. So a rooted tree has a root that denotes a common ancestor, while an unrooted tree only specifies the degree of relationship about taxa but not the evolutionary path like we had seen earlier. So for example, here in this unrooted tree we could see that this taxon A is more close to taxon B and C is more close to taxon D, while we can't tell from this tree about which taxa evolved from which. So also the branches of these trees could either be scaled or unscaled. So in scaled branches, the length of the branch is proportional to the evolutionary change. So that's called a phylogram. While in unscaled branches, the branch length is not proportional to the number of changes. So that is called a cladogram. So to define a tree, the number of leaf nodes, that is, the nodes that have no children or descendants, are given by L. And the internal nodes, that is, the leaves with branching nodes, is given by I. And the edges, that is, the branches, are given by E. So in this rooted tree, we could see that we have 3 leaf nodes, 2 internal nodes, and 1, 2, 3, 4 edges. So the total number of edges in a rooted tree is given by the formula 2L minus 2. So if you just calculate this, so 2 multiplied by L, the number of leaf nodes is 3. So 2 into 3, 6 minus 2 is 4. So which equals to 4 edges, as we could see in this tree over here. So this is a formula used to calculate the number of edges in a rooted tree. Similarly, for an unrooted tree as shown here, the leaf nodes are colored green and the internal nodes are colored red. So the number of edges here is given by the formula 2L minus 3. So in this tree here we have 2 into 1, 2, 3, 4 leaf nodes. So 8 minus 3 is 5 edges. 1, 2, 3, 4, 5 edges. So this is just a simple example of how we define a tree. So based on combinatorial calculations, the total number of unrooted trees that can be constructed using n number of samples or taxa is given by the formula 2n minus 5 double factorial. So now what's a double factorial? So the double factorial is the product of all the positive integers from n down to 1, taking every second integer. Like for example, a factorial of 5, that is 5 factorial is equal to 5 into 4 into 3 into 2 into 1, while a double factorial would be 5 into 3 into 1. So we just skip every second integer. So that's what a double factorial is called. Similarly, the total number of rooted trees that could be constructed for n number of samples is calculated by multiplying the total number of unrooted trees with the total number of edges. We just saw that the total number of unrooted trees is given by the formula 2n minus 5 double factorial, and the total number of edges, E is equal to 2L minus 3. So 2n minus 3 in this case. So solving this, we get the formula 2n minus 3 double factorial. So this is the formula used to calculate the number of rooted trees. So here is a summary of the number of rooted and unrooted trees for up to 10 samples using the formula we just saw. So since all of these numbers are hard to remember and it gets difficult for higher number of samples, you can't manually calculate it, it's worthwhile to remember the formula we just saw. So with that said, coming back to our question, number of unrooted trees in a phylogeny of 5 sequences is 3, 15, 105 or 945. So the keywords here to note are unrooted trees and 5 sequences. So n is equal to 5 here, and we need to calculate the number of possible unrooted trees that could be constructed. So for 5 sequences, the number of unrooted trees can be calculated using the formula 2n minus 5 double factorial. So here n is 5, so 2 into 5 is 10, so 10 minus 5 is 5. So here we calculate the double factorial of 5, which is 5 into 3 into 1, it's 15. So there are 15 different unrooted trees for a phylogeny with 5 sequences, which answers our question. So the answer is option B, 15.